The last three cell types that we're going to discuss in this set of notes are all examples of phagocytic cells. So just a recap on what phagocytosis or phagocytic cells can do. We briefly talked about these cells when we were looking at apoptosis. So these particular cells will engulf a pathogen or a non-self antigen via endocytosis. So we can see endocytosis happening here in step 1. The pathogen is being engulfed via endocytosis. Once that pathogen or non-self antigen is inside this phagocytic cell, that phagocytic cell naturally produces different digestive enzymes that will interact with the pathogen or non-self antigen and actually begin to break down those different components so that they are no longer functional, they're destroyed. And they can then release that waste product from the cell. Our neutrophils are the first type of phagocytic cell that we're going to look at, and the reason I put them first is because they are the most common type of phagocytic cell. So these are the ones that will be the first to any sign of infection. So as soon as your body detects that there is a non-self antigen or a pathogen that has invaded your body that's got past your first line of defense, these guys are going to be the quickest to respond and get to that site of infection and try and stop that pathogen from taking hold in your body. And the reason that they're so quick or the first to arrive at the site of the infection is because these are the phagocytic cells or your white blood cells that are actually circulating in your bloodstream. Okay, so not every white blood cell will be circulating in your bloodstream, but these are one of those white blood cells that will be there. As we discussed on the previous slide, they are a type of phagocytic cell, so they will engulf any pathogen or non-self antigen that they detect by using endocytosis and then the enzymes within them to digest those components. Our second type of phagocytic cell is known as a macrophage. Macrophage perform a pretty similar function to the neutrophil. Couple of differences to point out. So the first one being these ones don't circulate in your bloodstream, so they're not as quick to the site of infection. They're found in the tissues. So obviously it takes a little bit longer to travel through your tissue than it does flowing in your blood. So they won't arrive to the site of an infection or a pathogen or a non-self antigen entering your body until a little bit later. They are also a type of cell that we are going to focus on in a little bit called an antigen presenting cell. So this particular phagocytic cell, so the macrophages and also the dendritic cells, fall into the category of being antigen presenting cells, and I'll talk to you guys a bit more about that in a moment. So for now, macrophages are phagocytic cells. When they do interact with a pathogen or a non-self antigen, they will perform phagocytosis and engulf that pathogen or non-self antigen to hopefully destroy it using those digestive enzymes inside them. And these ones are found in your tissue, not your bloodstream. So they are very localized. They can't just, hijack your circulatory system and be whizzed around your body. They very much will only act on the areas where they are located. In saying that, they are located all over your body. So you will have macrophages fighting any sort of infection, no matter where it is. And our last type of immune cell that falls into the category of second line of defense is our dendritic cells. Pretty similar function to the last two types of phagocytic cells I've spoken to you about. Probably the key difference with these guys will be, one, they're found in the tissue of your body, so not in your bloodstream like the neutrophils. Two, they are a type of antigen presenting cell, which we'll talk about in a moment. And three is their distinct shape. Okay, so these guys have lots of like little tentacly arm thingies sticking out of them, which is really handy when they're trying to, I guess, almost stretch out and reach for that pathogen or that non-self antigen that might be a little bit further away. So I guess that's one little advantage that they do have. But I'm sure you would have noticed with those images that none of the other immune cells that we've looked at today have these little, tentacle sort of structures coming out of them. Still performs the same function as our other phagocytic cells in terms of will engulf a pathogen or a non-self antigen, use digestive enzymes to destroy it, and then hopefully prevent any further infections from developing. And the last thing that we're going to have a look at is this concept of antigen presenting cells. So this only applies to the macrophages and the dendritic cells that we've spoken about today. It doesn't apply to the natural killer cells or the neutrophils or the mast cells, just the macrophages and the dendritic cells. So these guys have the special ability to, when they capture a pathogen and they engulf it via phagocytosis and use their digestive enzymes to destroy that pathogen, they actually have this special ability to hold on to some of the pathogen's antigens and display them on their MHC Class II markers. Now, in the previous set of notes, we discussed the idea of an MHC Class I marker and a Class II marker, but we mainly only focused on a Class I marker. So just to quickly remind you of what the difference was. So we've got our cell here. It belongs to our body. So obviously this cell needs to have some self markers on top. Okay, so these little bumps here are going to be my self markers that will indicate to this organism that this cell belongs in its body. Now these self markers we can also call them self antigens, and we can also call them MHC Class I markers. These were the markers that all cells in your body except for your red blood cells had on their surface to allow your body to detect them as belonging to your body. So all the immune cells that we've spoken about today have MHC Class I markers. Macrophages, dendritic cells, natural killer cells, mast cells, neutrophils, all of them, they have MHC Class I markers. Macrophages and dendritic cells also have MHC Class II markers. Okay, so they've got extra set of markers on the surface of these cells. All right, so I'm just drawing them in purple now to illustrate that they have this second type. MHC Class II markers. And the purpose of these markers is when this antigen presenting cell, so a macrophage or a dendritic cell, engulfs a pathogen or a non-self antigen, it uses those digestive enzymes to break it up, but it holds onto a very small part of that pathogen, a part of its antigen, and it actually places the antigen of the pathogen and attaches it to an MHC Class II marker. So I'm just switching my pen to green so that you can see it. So it's digested, say, a bacteria, and this bacteria has green little circle antigens on it. After it's digested it, this particular antigen presenting cell will hold on to some of those little green circular antigens from the bacteria and present them or place them on the surface of the MHC Class II markers. And the function of doing that is so that these antigen presenting cells can take this message or take this non-self antigen to the more specific cells of your immune system. So the cells in your third line of defense or your adaptive immune response that have a really specific targeted role, are much more powerful at fighting off pathogens as well. And by taking this message or this non-self antigen to those cells in the third line of defense, it can hopefully trigger your third line of defense to kick into action and help protect your body against this pathogen at a much faster rate. We're just going to flick over to the next couple of slides, and hopefully the animation works as I'm recording my voice on top of it. But essentially the animation is just trying to show you what happens when a macrophage or a dendritic cell engulfs a pathogen, and how that antigen that was on the surface of the pathogen can end up on an MHC Class II marker. Okay, so as you can see on the screen here, we've got some pathogens in orange here that have some non-self antigens on their surface. So my body, your body, they would recognize those antigens on the surface of these pathogens as non-self. So if one of our immune cells were to come into contact or come across the path of these pathogens, they would hopefully recognize them as non-self. The blue image that is coming into the screen now is an example of a phagocytic cell. So you can see that it's engulfing those pathogens because it has recognized them as having non-self antigens on the surface. Let's have a look at what happens next. So now we're having a look at the inside of our phagocytic cells. So our phagocytic cell is shown in blue, and we can see that it has engulfed our pathogen, which had non-self antigens on its surface. Okay. Oops. Now what you're seeing on the screen now is a very simplified version of what happens when this particular phagocytic cell, so either a dendritic cell or a macrophage, digests the pathogen. So we saw the orange part of the pathogen sort of disintegrate away, and it left behind some of the antigens from the pathogen's surface. And now we're also seeing these MHC Class II markers in grey, and they're carrying those non-self antigens that belonged to the pathogen, for example a bacteria, to the surface of the cell. So those MHC Class II markers are now on the surface of the cell. So this bit's the MHC Class II marker, and this little pink triangle bit was the non-self antigen that belonged to the pathogen. Same over here on the other side of the image, we've got two MHC Class II markers, each one with its own non-self antigen that it is presenting on the outside of the cell. Now the goal of the antigen presenting cells is to obviously present these non-self antigens to the more powerful or the more developed immune cells of your third line of defense, so your B and T cells. They are not cells that we have discussed yet. We're going to discuss them in a later set of notes. So obviously we'll revisit this idea of being an antigen presenting cell and why that is so important.